Alright, so we're starting up now with our fourth section on enzymes and energy, and we're continuing on now with the idea of catalysts, just in general, and then we'll be moving into a more specific approach onto what enzymes are and how exactly they do what they do. So a catalyst is really any substance that can speed up the rate of a chemical reaction. Now, it's important to realize that catalysts and enzymes are just one type of catalyst. They're biological catalysts, specifically enzymes. Catalysts don't ever get you more product from a reaction, but they get you the same amount of product faster, as if you didn't have a catalyst, right? So you get to the end point quicker than if you didn't have a catalyst. And I talked before about a section, or in one of the earlier sections, about a race with two different cars, and the finish line's in the same place, but if you're driving a faster car, you get to the finish line quicker. Same idea with a catalyst. If you have a catalyst, you get to the same finish line as if you didn't have a catalyst, but you just get there a lot faster. So, one good example of that, this is a completely non-biological example of a catalyst. A little bit of a story here. It goes back to, I guess we'll start back in like the late 1970s, when cars actually used leaded gas. And the reason why they used lead in gas was to make the engine run smoother and to make it so your engine didn't knock as you were driving. So pretty quickly they found out that lead in gasoline was not the best thing to have because you were burning it, it was going into the atmosphere, and people, especially kids, were ending up with all kinds of neurological problems. So by the mid, let's see, mid to late 1980s, they started requiring that leaded gas be no longer a thing. And instead of lead, they started putting octane in gas. Now if you go to the gas station, you see octane, right? So you get 87, 89, 93, whatever octane. And so octane replaced lead as the anti-knock agent. The problem with that is that if you burn octane, you also get carbon monoxide. And of course, most of us know carbon monoxide is also poisonous for a very different reason than lead, but it's also a very nasty, harmful chemical that if you're exposed to large amounts of it, like if you live in a city, for example, and there's all kinds of car exhaust around, you can actually come up with carbon monoxide poisoning. So, what they eventually ended up doing by the early 90s was mandating that all new vehicles be sold with a catalytic converter. And so a catalytic converter is essentially a way to prevent at least big amounts of carbon monoxide from coming out of the car's tailpipe and going into the atmosphere. There's still some, but nowhere near as much as what there was before the catalytic converter was mandated. So what happens there is that when octane is burned, the carbon monoxide reacts with oxygen and a metal in the catalytic converter to produce carbon dioxide and also heat energy. And so the catalyst in this case is actually platinum. Platinum is the stuff in the catalytic converter that causes this conversion of carbon monoxide and oxygen into carbon dioxide, which is a much less, at least biologically harmful substance than carbon monoxide is, right? So it speeds up the conversion of carbon monoxide to carbon dioxide and therefore makes it less harmful. So this actually speeds it up by quite a bit. So this is the idea again. We talked before about activation energy in exergonic reactions, and if you have a reaction that requires a large amount of activation energy, it's going to take a bit longer. But if you catalyze a reaction and reduce the amount of activation energy you need, that makes the reaction go a lot faster. Now again, you're getting to the same endpoint, you're producing the same amount of products, you're just doing it a whole lot faster with a catalyst because the activation energy is less when you have the catalyst present than if you don't have the catalyst present. So enzymes then are biological catalysts. I mentioned this before. They are proteins. And just like any other protein, our cells have genes that tell them how to make proteins, in this case enzymes. And so, for example, if a cell has an incorrect version of a gene for an enzyme or is lacking a gene for an enzyme, that can have some pretty big consequences for that cell in that the cell cannot produce the proteins it needs to actually do the reactions it needs to do. So a good example of this again is lactose. Lactose is a disaccharide sugar that we talked about a couple chapters ago. It's made up of glucose and galactose, and lactose, of course, is milk sugar that's present in most dairy products. And it's really not a problem to break down. Our bodies don't do it real well unless they happen to produce the enzyme lactase. And lactase is an enzyme produced by certain cells in our small intestine. As long as they're making lactase, your body can break down lactose, you get your glucose and galactose out of it, no problem. No issue with lactose intolerance, no side effects, no symptoms, all is well. You get the energy out of lactose and you're good to go. The problem with that is that our bodies don't always produce lactase, and we'll get to that here in a second. Now, one thing to think about is, okay, what happens if we have a milk jug sitting on the shelf at the grocery store? Now, that milk has been pasteurized. It has to be. Legally, it has to be pasteurized to be sold in a grocery store. So, pasteurization is essentially a heating process that kills off any bacteria that might be in these dairy products so that they don't make us sick when we eat or drink them. Um, so, by killing off the bacteria, the milk stays relatively stable as long as it's refrigerated on the store shelf for a couple of weeks, probably, before it starts to go bad. And you might wonder, well, why does it start to go bad if it was pasteurized? Well, it turns out the milk actually has resting stages of bacteria in it that survive the pasteurization process, and they they can actually essentially start to grow even if they're refrigerated after a couple of weeks. They start to grow again, and that can actually cause the milk to go bad because the bacteria produce the lactase. The lactase, when it's broken down in anaerobic conditions, actually produces the bacteria produce an acid in the process, which causes the milk to curdle or spoil, right? So that's kind of the idea there. Now, as far as lactose intolerance, the idea there is that as we age as mammals, most mammals, once they reach a certain point, get to a point where they stop drinking milk from their mothers. And so it makes sense then for their bodies to shut off their genes for lactase production. Humans are unique and kind of strange in the sense that a lot of times we continue to consume dairy products even after we're infants, well into our adult years. And not only that, but we consume milk from different species, which is kind of weird as well if you think about it. But the problem with this is that our bodies are still programmed to shut off the gene for lactase production as we get older, even though we still want to consume that lactose in those dairy products, right? So what ends up happening is that we consume the lactose, our bodies don't break it down because we're not making lactase, and that milk sugar eventually makes its way to our small intestines, and then eventually our large intestines where there are bacteria living. Things like E. coli bacteria, which live in our large intestines, are actually very important for us. The problem with lactose, though, is that these bacteria ferment lactose in anaerobic conditions in our large intestine, and one of the byproducts of that is carbon dioxide gas, and that gas is actually what produces these symptoms of lactose intolerance if you happen to suffer from those symptoms. So the Cliff Notes version of that is that our bodies stop making lactase. It makes us harder for our bodies to break down lactose, so therefore the bacteria break it down instead. The bacteria ferment it, and they produce gas as a byproduct, therefore lactose intolerance, right? All right, so a little more about enzymes here. We talked before in our previous section of lecture about how most of the reactions that happen in our bodies wouldn't happen fast enough at body temperature to really be able to sustain us as living things. So enzymes make it so these reactions happen much, much faster, even at body temperature, therefore keeping us alive. Now, one of the other important things about enzymes to realize is that the enzymes themselves are not consumed by the reaction they catalyze. The enzymes are actually recycled, or I guess reused technically over and over and over again, such that you might have one molecule of an enzyme doing, like the slide says, maybe 10,000 reactions per second. That's just for one molecule of enzyme. So the enzyme will catalyze a reaction, let it go, catalyze a reaction, let it go over and over and over and over again without actually being consumed. So in a cell, you might only need a few molecules of enzyme to do all the reactions that that enzyme needs to do. And so, like we said before, enzymes are proteins, and like every other protein in our cells, we have to have genes that tell our cells how to make those proteins. If you're missing the gene, or if the gene's in an incorrect form, the enzyme is either not produced or it's produced incorrectly, and therefore it doesn't really work as well as it should. So no gene equals no enzyme. So getting more into how enzymes actually function, enzymes work by pre-stressing the bonds in the molecules they're working on. So whenever we have an enzyme catalyzed reaction, we don't really call your starting ingredients reactants anymore. We call them substrates. So substrates are what you begin with in an enzyme catalyzed reaction. So you have your substrate, you have your enzyme, and eventually you get your product, right? So what the enzyme is doing to the substrate, like I said, is pre-stressing the bonds, making the bonds easier to break, therefore lowering the activation energy of the reaction. So in contrast to non-biological catalysts, enzymes are very, very specific. Now, most enzymes will only work on one or maybe two very, very similar substrates. Otherwise, they won't work at all. It's very much like a lock and key mechanism where the lock is the enzyme and the key is the substrate. So there has to be a very much complementary fit if they're going to work together to actually do the reaction, right? The other thing that can happen with enzymes is we have a lot of different ways to control their activity. And we're going to get to those. That's kind of like the last section of this whole chapter is talking about how enzyme activity is controlled. either through competitive inhibition or non-competitive inhibition or feedback inhibition, all different ways of controlling this. We're going to get to all of those. But one of the things that happens here is that as the products of an enzyme catalyzed reaction build up, they can actually slow down further reaction from occurring, which really makes sense. It's a really handy way to kind of conserve your substrates. Is once you have enough product, you don't want to keep making more of it, so that product itself actually interferes with further reaction and therefore helps you conserve your substrates. So these enzymes, like we said, usually are proteins, sometimes RNA as well. There is a very temporary fit and sort of bonding of the substrate to the enzyme temporarily to form what's called an enzyme substrate complex. So really, if you're looking at an enzyme catalyzed reaction sort of generically, what you actually start off with is your substrates plus your enzyme, and that gives you what's called an enzyme substrate complex, where they're actually bound together. Like the key is actually fit into the lock temporarily. That's the enzyme substrate complex. And then eventually you get your products, and then also your enzymes. Remember, because your enzymes are reused after every reaction. So you start off with substrate and enzyme, you get an enzyme substrate complex, and then eventually get your products, and you get your enzyme back again to do some more reaction. But while the enzyme is bound to the substrate is being weakened. And the weaker it is, the less activation energy you need to eventually break it, and therefore the faster that reaction is going to go. So a really good example of this, this is just for illustration. You don't certainly have to know the numbers here, but carbonic anhydrase is a really important enzyme that we have in our body, in our blood especially, to help speed up the conversion of CO2 and water into something called carbonic acid. And then carbonic acid breaks down to form bicarbonate ions and hydrogen ions. This is all part of the blood buffering system. We talked about it back at the end of chapter 2. But getting this carbonic acid formed is a really important way to help our bodies transport carbon dioxide in our blood. So it turns out that if you have carbon dioxide in contact with water, it will form carbonic acid naturally, even without the enzyme. But it does so very slowly. And this is the reason why rainwater is slightly acidic, right? Because we have CO2 in the air, we have water vapor in the air, they mix together, they form a little bit of carbonic acid, therefore rainwater is slightly on the acidic side of the pH scale. But if we throw in carbonic anhydrase, which is an enzyme made by cells, then all of a sudden we go from 200 molecules per hour to 600,000 molecules per second with the enzyme, which is a pretty dramatic rate of increase of reaction when you have the enzyme versus when you don't. So, when I say the enzymes are speeding up the reaction, we're not talking about making them twice as fast or 3 times as fast. We're talking about making them thousands, tens of thousands, millions of times faster with the enzyme versus without the enzyme. Now, a really important part of the enzyme is something called the active site. And the active site is the part of the enzyme where the substrate actually fits in so that it can be weakened and then eventually released and eventually finish out the reaction. So the active site is where the enzyme substrate complex is going to form temporarily and the activation energy is going to be lowered. And again, like I said before, this is complementary fit. The substrate is going to be the key, the enzyme is going to be the lock, and specifically the active site is the precise position on the enzyme where the key fits into the lock complementarily, I guess that way. So this allows for the bonds in the substrate to be stressed, the activation energy lowered, and therefore ultimately the rate of reaction to be increased. So this is an example of an enzyme called sucrase. This is sucrase here. This guy up here is sucrose. So I think in this picture the green is going to be glucose and the orange is going to be fructose. Yeah, that's right. So glucose and fructose, 6-carbon sugar, 5-carbon sugar. This is a different shaped 6-carbon sugar. It looks like a 5. There's actually a 6 one coming off over here somewhere. So we have sucrose here, glucose and fructose, bonding onto sucrase. We get that temporary fit, that enzyme substrate complex forming, and if you notice right here in the middle, what's happening to this bond? This bond is being stressed. It's being pre-weakened by the fact that it's present on this enzyme. And so this bond breaks very easily. Glucose and fructose are released. Now our bodies can take advantage of those. We can burn glucose, we can burn fructose, we can use them for energy, just like we're kind of set up to do. And then our sucrase enzyme is also released back to the point where it can be used to catalyze another reaction. So it's being reused like we talked about before. So that's a pretty good, fairly basic example of what an enzyme catalyzed reaction would look like. We have our substrate, we have our active site on our enzyme substrate complex, we have our products, we have our enzyme back, and the reaction just keeps repeating. All right, so these enzymes, they can be in various places in the cell. They can be in the cytoplasm, they can be associated with various organelles, They can be built into the plasma membrane, which is a really important point if you're dealing with a prokaryotic cell that doesn't have a lot of organelles, or really any organelles aside from ribosomes. They use their plasma membrane for just about everything, right? So the enzymes they need to do all the kinds of things that we do with our organelles, they have built into their plasma membrane, and that's actually where they do the bulk of their reactions. Some enzymes, as it says, also require coenzymes. They can be various types of inorganic things, metals. They can be vitamins, for example, which are kind of like enzyme helpers. The enzyme by itself, without the coenzyme, depending on which enzyme we're dealing with, may not be enough to actually run the reaction. Sometimes you have to have the coenzyme there as well for the reaction to work. And a lot of times, too, we see what are called multi-enzyme complexes, where we'll actually have several enzymes kind of together, so that when the product of one reaction comes out, it immediately goes into another enzyme and runs through a second reaction immediately. So the product doesn't have to travel very far before it hits the next enzyme in the process to actually do the next reaction. So that's actually a way to kind of increase efficiency as well. As far as ways of affecting enzyme activity, and we're going to get through this kind of quickly, and then we'll wrap up this section and probably start on our final section from there. Remember, enzymes are proteins. And back in one of our chemistry chapters, we talked about different types of environmental conditions that can affect protein structure, right? And we said that protein structure is everything for a protein's function. If you don't have the correct structure, you're not going to have the correct function. If you change the structure of a protein, you change the function of the protein. And so anything that affects protein structure can then potentially affect a protein's function, in this case an enzyme's function. So if we have an increase in temperature especially, that can cause proteins, in this case enzymes, to denature and therefore stop functioning. If we have a change in pH, if the environments of, say, the blood, if the blood pH, which is normally between about 7.3 and 7.4, if it were to go below 7.3 or go above 7.4, that carbonic anhydrase enzyme we talked about in the blood could start to denature, and that could very much affect our body's ability to carry CO2, which could be catastrophic if it's a big enough change. So pH can also cause denaturation of enzymes. Salinity or ion concentration can also change protein structure, enzyme function in this case. And so one of the reasons why our bodies need to maintain homeostasis for so many different kinds of things like temperature and pH and ion concentration is so that we can protect our enzymes in our body and therefore maintain the chemical reactions that our bodies need to stay alive. So we're actually protecting enzymes in the process of maintaining homeostasis. The coenzyme concentration as well. For some enzymes, you can have as much enzyme as you can imagine. If you don't have the coenzymes, the reaction is just not going to work the way it's supposed to. So very, very important, but also very delicate in how they function. All right, so different organisms have different optima for various types of conditions. This is a pretty good example of that shows it really well, I think. The top picture up here is contrasting a human enzyme versus a hot springs prokaryotic enzyme. And what we see here is the optimum for these two enzymes is very, very different. For the human enzyme, the optimum is right around 38, 39, 40 degrees Celsius, right around body temperature. And for the hot springs prokaryote, the bacteria, it's around maybe 72 or 73 Celsius. So neither of these two enzymes, the human enzyme would be absolutely obliterated at 73 Celsius. And the same thing really for the prokaryotic enzyme. They don't really work at each other's optimum temperatures. Okay? There is no blue enzyme function over here, and there's no red enzyme function over here, because if you change the environment of either enzyme, they're completely denatured and broken down. They're just not going to be present anymore at that point. Same thing for pH. Right? Pepsin is an enzyme we find in the stomach. Trypsin is an enzyme we find in the small intestine. Very, very different environments. Despite the fact that the stomach leads into the small intestine, there's a really big change in pH as you go from the stomach to the small intestine. So either one of these two enzymes placed in the other's environment would denature and stop functioning. So really important to have the right environmental conditions for these enzymes. All right, so we're going to end this section here, and we're going to pick up with the next section here in just a moment.